Since I see a few people still joining, we wait for 3 to 4 minutes more and then we begin with our session. Thank you. Hello everyone. I would like to welcome you all to this session of SCIOMICS Club. Before starting with today's session, I would like to brief you all about who are we and what do we do. SCIOMICS Club is a bioinformatics and data science student-led club that originated in India. Our main goal is to help students learn about bioinformatics, gain its practical knowledge through group sessions, debates, easy to take courses, and ultimately push their limits of the students beyond textbook limits and push their critical thinking limits as well. We also aim at encouraging young minds to push their critical thinking limits beyond the school and college curriculum. Now, I'm Rutvi Vaja, president of SCIOMICS Club. I am a student of integrated biomedical sciences, and if anyone is interested to join the club, they can join the club by the following this WhatsApp group link, or else you can even email me on the email ID stated over here. Also, we do not have any age restrictions for joining the club, hence we welcome all ages because we believe that knowledge has no boundaries. What do we do in SCIOMICS Club? So we have interactive sessions on Zoom in which we interact with people from various backgrounds. We also have certain kinds of groups and debates. We have certain kinds of OmicsLogic training programs that comprises of introduction to bioinformatics, genomics, metagenomics, transcriptomics, and a very new course introduced that is bytes and molecules. So we understand biological literature in such a way that it would help us to put that exactly into our practical lives. Moving on, the topics that we cover in SCIOMICS Club are about mainly focused on infectious disease, oncology, neuroscience, plant science. We also have topics related to genomics and microbiology. Our main aim is to help students to clear concepts about bioinformatics. How can they use tools about hands-on directive sessions is also what we conduct. We have brainstorming sessions that help the group of students to enhance their critical thinking abilities. And also, we have certain kinds of programs that have shown applications of coding in bioinformatics. So, if students are interested in that, we also provide that. In order, SCIOMICS Club aims at helping students learn bioinformatics from ground zero. We also provide free courses on OmicsLogic and Pine Biotech platform, namely Introduction to Bioinformatics and Introduction to Genomics. They are self-learning introductory courseworks and anyone can take them. They are available for free on the website. So what do we usually do in SCIOMICS Club? We have group discussions. We have review report writing on certain kinds of biological literature in order to apprehend them well. We have debate on certain kinds of mind-boggling arenas in bioinformatics comprises of like waxing, bioinformatics and technology and further on. We also have presentations on certain crucial research topics and research papers. Our research interns who are doing projects with Pine Biotech have also been asked to present in SCIOMICS Club. We also discuss and exchange ideas for project development, hence students can take their ideas further and work on a hands-on project. So now I would like to, this session is currently based on the theme cancer biology. Today, our research interns and our mentors, including and also I, would be presenting upon certain themes that are based on oncology and the ongoing projects. To present first, I would like to invite Sreyasee Banerjee and Sraosee Banerjee, who are doing the project based on cancer biology, that is breast cancer, as a team. To start with, Sreyasee Banerjee and Sraosee Banerjee both are from West Bengal, currently pursuing their masters in biochemistry from Rewa University, Bangalore. Sreyasee is interested in bioinformatics and its application in the drug development field, and Sraosee is interested in bioinformatics application and its use in drug development, also for cancer and its treatment. I invite Sreyasee and Sraosee Banerjee to please share their screen and take their presentation forward. Thank you.
Yeah, I'll be sharing my screen right now.
Yes.
So are we seeing something? I don't see any slides being shown.
It's taking some time. Okay.
Try resharing it.
Yeah, in case you have a problem, Rutvi, you can also share the screen, right?
Yes, I can share the screen for them.
Okay, I think you should start share the screen. And Sreyasee and Sraosee, you can then continue with the presentation.
Yeah, sure.
Okay, thank you, Rutvi.
Is my screen visible?
Yeah, it's visible.
Yeah.
Yeah, so this is me, Sreyasee Banerjee. I'm a student of Reva University, and currently I'm pursuing my final semesters in master's degree in biochemistry. And
here I'm Sreyasee Banerjee, currently pursuing a master's in biochemistry from Reva University. I did my bachelor's from Utkal University.
and now Sreyasee Banerjee will be explaining some main points for breast cancer.
Now, what is breast cancer? It is breast cancer is the most common malignant condition of breast. After skin cancer, breast cancer is the most uncontrollable, most common cancer that is diagnosed in the United States. The causes mainly for the breast cancer can be hormonal factors like when cells divide more rapidly than healthy cells do, and then they continue to accumulate, forming a lump or mass. Cells may spread through the breast to our lymph nodes to other parts of the body. There are different types of breast cancers, that is ductal carcinoma in situ, invasive ductal carcinoma, inflammatory breast cancer, and metastatic breast cancer. Now, the risk for the breast cancers like passive smoking releases benzopyrene rings that causes breast cancer in women. Then radiation treatment to the chest as a child increases the breast cancer. Hereditary, having dense breast. Symptoms can be a lump in the breast or in underarm, thickening or swelling of part of the breast, irritation of breast skin, then changes in the size or shape, blood discharge from nipples, and also newly inverted nipple, and morphological changes in the organ. Okay, next slide. So now coming to the mutations that causes breast cancer, that can be BRCA1 and BRCA2. These are the most common cause of hereditary breast cancer that is inherited. These are the inherited mutation in the BRCA1 or BRCA2 gene. In normal conditions, these genes help to make proteins that repair the damaged DNA. Also, the mutated versions of these genes can lead to abnormal cell growth that leads to cancer. Also, the another gene mutation that causes breast cancer is mutation in P53. The frequency of this is very less. Now, Sreyasee will be explaining the next bioinformatical approach for breast cancer.
Yeah, so in bioinformatical approach, what we do is we compare unknown sequences of DNA from a collection of samples, and then we compare with the control, and we identify the mutated sequences, and elucidation of the expression profiles are done. That is mainly for breast tumorigenesis, and through bioinformatical analysis, this can be achieved. Also, high throughput genomic technology nowadays has become very rapid and major tool for the study of breast cancer. The gene expression profiling has been applied to many areas of research, from basic science to translational science, and also to identify targets and treatments for them. And gene amplification occurs, and also like high throughput genomic technology enables us to see breast cancer from a bioinformatical point of view, where we can we can analyze the data set, and we can identify the markers, the sites where the mutation occur, and according to it we can devise our biomarkers or the therapeutic drugs. So next slide please. Yeah, following transcriptomics and proteomics, the large scale and high throughput techniques help us to do further treatments and prediction of the outcomes. These include like SESTs, SAGE, MPS. These are a part of the techniques, and the gene expression profiles analysis, the differential expression of the genes. Differential expressed genes are like those have a difference or change observed in their expression profiles during two experimental conditions. One when is a mutated condition and another in normal conditions. And also hub genes, I wanted to say that hub genes are the genes with higher correlations. So in breast cancer, the most correlated genes, we can tell them as hub genes. And by identifying the hub genes, we can identify the biomarkers, and and following that path, we can identify the sites through which the therapeutic drug can be devised. So next slide. Yeah, so after we identify the changes, the mutated sequences, and the analysis that we will do, then the biomarkers and therapeutic drugs will be unleashed, and they are enabled due to the presence of bioinformatical studies, definitely. And molecular classification of the breast cancer can be done by various microarray analysis. And by using bioinformatical approach, treatment and drugs for the breast cancer nowadays has become very helpful, and in very less time it is solved right now. So yeah, that's it. Thank you.
Thank you for your insightful information. I may throw it open to the audience. If you have any questions, please put it down in the chat box so they can answer.
Hi, Rutvi. Vishwa has a question.
Yes,
So,
please.
Sraushi, can you take this question? Thank you.
Yes, ma'am. Chances of breast cancer in male is very low than females, because the mammary glands are affected, which are not developed in males. So chances are quite low in males for developing breast cancer, and it is mostly affected to females. So am I clear, or I should explain more? Okay, thank you.
Thank you once again for presenting. Now we will move on to the next presenter, that is me. I'm Rutvi Vaja, and I would be presenting on two topics today. The first one is precision medicine. I would be presenting on precision medicine, that is how it is useful. And as we have the theme for cancer biology, I would be explaining it with the example of skin cancer. And further on, I would also explain like about 5 to 6 slides about my current present topic, that is medulloblastoma cancer project that I'm working on. So first we will see about precision medicine. So precision medicine is also known as the personalized medicine, which is taking over the industry of molecular medicine and bioinformatics. And that's why we call it the next generation's future. To get familiarized with what personalized medicine is, personalized medicine is a term that we use to describe the individual treatment a person gets according to his or her genetic makeup, that is genotype, the phenotype that they express, the biomarkers, and the psychological characteristics that they possess. So personalized medicine isn't kind of a medicine that is given according to your own characteristics or the traits that a person prevails, and it is only particular for that kind of an individual, and one personalized medicine could not be taken by second individual because individuals differ from each other in terms of genotype, biomarkers, and several other traits. So personalized medicine is a whole branch that is prepared from huge clinical data and bases that comprises of combining the genomic profile, the proteomics profile, the transcriptomics, epigenetic, and microbiome profile of an individual, upon which the conclusions are elucidated, and later on the personalized medicine for that individual is made. To get more into depth about what personalized medicine is, branches like phenomics, genomics, transcriptomics, and proteomics, epigenomics, and microbiomics, which are all the sub branches of bioinformatics, are included into precision medicine, which together makes one unit. So, if there is a population of many, I hope you can see my cursor. For a more better and personalized care for a healthy individual, we have to give the treatment one-on-one, that is for treating one individual. This would increase the population levels of health in terms of care, the kind of health we provide, and kind of the healthy nation that we prevail through. There is a better risk stratification, that is risk can be minimized, errors can be minimized, and results can be greatly improved with the use of precision medicine. Though precision medicine is not still widely accepted and used, but however we have found very much success rate in precision medicine in different eras of science, molecular medicine, and biology. So why do we go to personalized medicine instead of the regular medicine that we go for? So there are various reasons that stands out. First is it improves disease detection. Like personalized medicine is so apt that it can detect a disease according to your body type, the kind of activities you do, your characteristics, and further on. And hence it is able to give an effective treatment according to your genotype, which is very important point in itself. It also helps us to shift the emphasis in medicine from reaction, because normal medicines we take, we wait for it to get cured. But over here, we can go to one step further. That is, in personalized medicine, we need to see the reaction, but what we actually experience is prevention. We can also customize disease prevention strategies, like they advise us on what food we have to eat, the diet we have to follow, the practices we have to go through while we are on personalized medication. It also reduces the time and cost of the nation that ones have to spend beyond other kinds of medicines. And we have also decreased rate of clinical trials that everyone has to go through, and we all know this in times of COVID-19 pandemic. We can also improve predict susceptibility to diseases. Like susceptibility to diseases is sometimes very hard to detect, like it takes 3 to 4 months and further on. But with personalized medicine, it is very easy to detect such kinds of diseases, especially cancer or melanoma or cardiovascular diseases, and further on. So we call it personalized medicine as a multi-phased approach. That is because the risk management is very much lower. We go through genetic screening and testing to reveal certain kinds of predisposition to these diseases. And the wonderful thing is that even before a person gets the diseases, if it prevails in hereditary, or if it doesn't even prevail in hereditary, one can get to know before the onset of the disease that if he or she is going to get this disease. So risk assessment factor is very low, and one could get preventive measures even before the onset of the disease. Second, as I already talked about, is prevention. The kind of behavior, lifestyle, and practices everyone, and the doctor especially, asks us to follow could be a great barrier in preventing the onset of that diseases. The treatment becomes as effective as we can go through, that I already discussed about. Diagnosis is also becoming accurate with the help of precision medicine. Further on, active monitoring, that is the management strategies of certain kinds of health and certain types of diseases which are not controllable right now could be possible with active management of precision medicine. So if you have taken the example of melanoma, that is screen cancer, in personalized medicine, like how do they work? So BRF is the human gene responsible for the production of the protein called as BRF. This protein is usually involved in sending signals inside the cells to direct cell growth, to form tumors and form mutations. So in 2011, scientists came across a drug that is known as vemurafenib, that is a BRAF protein inhibitor. So this test was later on approved for the treatment of late stage melanoma. However, around 60% of the patients have BRF mutation, and treating them with V600E mutation, that is the short form of vemurafenib BRAF protein inhibitor, was found to be successful. So this was my presentation about personalized medicine. I would like the audience to ask me questions so that I can move on to the second presentation. Thank you, Beepsa ma'am. Okay, just in case you have any question, please put it down in the chat box while I can start with my second presentation, because we have a time limit to follow. Okay, so my next presentation would be on medulloblastoma project that I have been working on since a week. It's just been a week since I started this project. So I am on my stage of, I'll just share my screen. Is my screen visible?
Yes, we can see your screen, Rutvi.
Okay, yeah. So I have been interested in working in medulloblastoma, that is the cancer of the brain. So first, just get started with medulloblastoma. It is a rapidly growing tumor of the cerebellum. The location of it is cerebellum, and in terms of kids, or maybe 10 years of children, it is in the vermis of the cerebellum. It occurs in the lower rear portion of the brain, and it is also known as the posterior fossa. So in some terms, it is also known as the cancer of the posterior fossa. It is located below the tentorium, that is a thick membrane which separates the large cerebral hemispheres of the brain from the cerebellum. In children, this cancer arises often near the vermis, which would be shown in the next slide with the picture depiction. So vermis is a narrow worm-like bridge that connects the cerebellums and the two sides together. In adults, this tumor occurs in the body of the cerebellum, especially towards the edges, and continues to grow further. Medulloblastoma is one of the fatal cancers which we have not been able to get a cure of until now. It is a type of primary brain cancer in children, and it originates in the part of the brain that is towards the back and the bottom, or the floor of the skull, which proves to be the cause that why it is very fatal. Over here on the picture of the right hand side, you can see the location of the tumors in the tentorium, the orange dots that you can see. Further on, this is the picture of the vermis, which shows that this is the vermis where the tumors occur in children. So tumors in children are present in vermis. There are certain kinds of histological types of medulloblastoma that are classified as classic medulloblastoma, demoplastic nodulomedulloblastoma, medulloblastoma with extensive nodularity, large cell medulloblastoma, anaplastic medulloblastoma, medullomyoblastoma, and melanotic medulloblastoma. Each of these medulloblastomas has their own characteristics and specifications. but all of them have proved to be fatal, and there are not enough disease preventions, and I must say biomarkers developed yet in order to prevent this or have any kind of healthcare prevention for this kind of disease. So what my exactly the problem question is, there have been works that have shown that cell cycle and p53 signaling pathway play an important role in invasion and progression of medulloblastoma from normal samples. There are certain kinds of genes also figured out, like the one stated over here, CCNA2, MAD2L1, and further on, that have been included to indulge in the invasion and progression of medulloblastoma. Now, what is actually P53 pathway? So P53 pathway actually plays a key role in controlling cell division and cell death, and it is also thought to prevent chromosomal replication during cell cycle. So if there is any kind of DNA damage or apoptosis, P53 gene activates the P53 protein in order to stop the replication of damaged DNA further on. So it arrests the cell cycle at G2 phase. So why we address this problem is because we want to find out the relation of miRNA and P53 protein pathway in medulloblastoma, which is largely unknown. We have to treat and identify miRNA because the nature of miRNA in all different types of medulloblastoma is different. So identifying the underlying mechanism beneath it would help to find certain kinds of therapeutic biomarkers as well. And how miRNA regulates the activity and function of p53 protein. It is thought to believe that miRNA controls p53 protein in various ways, that is by which miRNA interact with p53 and it network in multiple layers. Also, they activate and regulate the function of p53 through direct repression, which in itself is a problem. So further moving on, the three main types of questions and hypotheses I would like to address is, how does miRNA function in silencing genes and proteins? Like, is it by upstream regulation or downstream effectors? The second type of question I would like to address is, how does expression of miRNA differ in different types of medulloblastoma? Like, why do they react differently in different kind of medulloblastoma? Is there any aggressive state, or is there any normal states? Is that what I want to find out by this second question. The third question is to identify miRNA as therapeutic biomarker by examination of the role of p53 signaling pathway and cell cycle in development of medulloblastoma. Eventually, this forms my central question, that is the identification of therapeutic biomarkers. Now, there are several ways by which miRNA is also interconnected with p53 gene protein, but again, not much is known about this, and I think that if we indulge into this more, we can help at least someone to get rid of medulloblastoma, because it is very fatal and 95% of death is experienced with the persons who suffer to medulloblastoma. So coming about data sets, now I'm on the stage of my project where I have found data sets, but still the metadata analysis and everything is yet to be done. But I have found two data sets. One has healthy control samples, that is 6, another, and along with the medulloblastoma samples from exosomes and peripheral blood, that is 9 samples. I've also have got medulloblastoma samples from frozen tumor tissue. The another kind of data set that I have is with healthy control sample 6, medulloblastoma samples from cerebrospinal fluid, that is 25, and medulloblastoma samples from leptomeninges in metastasis state, that is 34 samples. Now I would like to further go on with this project by calculating the metadata, and further on I would like to work on this project as well. So my presentation ends over here. Can I have questions from you guys?
So I had a question. So what exactly do you mean by a therapeutic biomarker?
A therapeutic biomarker is like, over here I mean indication that a progression of medulloblastoma has taken place or is about to take place.
So you mean this is an early biomarker, so you can
Yes,
detect that
predictive
early?
biomarker.
Yeah, I wouldn't call that a therapeutic biomarker. Therapeutic biomarker means it's a marker of therapy, that a particular type of therapy will work.
Okay.
And you're more focused on a diagnostic biomarker to diagnose it earlier.
Yes, kind of predictive biomarker.
Harpreet, did you have any comments on this?
Hi, Rutvi.
Hello,
I
Harpreet
have
ma'am.
one question.
Yes.
So have you looked at exactly the diagnostic biomarker, maybe the candidates that are available already for the medulloblastoma?
Yes, I have gone through them, but I haven't included them in the slides.
No, I mean, when you're analyzing, I mean, have you looked at actually what are already miRNA or RNA biomarkers available, and what are their limitations?
Yes, ma'am. So I've analyzed the biomarkers, like especially miRNA biomarkers for medulloblastoma. The limitations are that they are able to show the progression of the biomarkers like MIR143, that is a predictive prognostic biomarker, but it can also act as an oncogene or tumor suppressors, but the success rate has not been calculated that further, so that's why they are even confused with whether to take this forward or not.
So there's another question in the chat. Do you see the chat, Rutvi?
Yes. How are you planning to analyze your data further? Which tools are you using? Okay, so for my data, I have the data sets over here I have mentioned are all single end reads, but I have another data set with paired end reads of miRNA with my sequencing. So that data I would like to analyze miRNA data so that I can get to know that if they have upstream regulation or downstream regulation, and later on I would analyze this data. But how would I go with this is not what I'm sure about, because I've just started with my project and it's not even a week, so I'm still exploring.
Are there any other questions for Rutvi? Then I think Rutvi, you should move on to the next presenter. Thank you.
Yes, so our next presenter is Clinton. He is our associate OmicsLogic mentor who conducts the sessions with oncology programs associated with Pine Biotech. He further has a great background in his own education level. He has done his Masters of Bioinformatics from University of Guelph. He has done his Masters of Cellular and Molecular Medicine from University of Ottawa, and he has done his Bachelor's of Biological Science from Carnegie Mellon University. I would like to hand it over to Clinton. You can further share on your screen and go ahead with your presentation. Thank you.
Thank you, Rutvi. I will share my screen. All right. So my name is Clinton Kuna. As Rutvi mentioned, I completed my bachelor's in Carnegie Mellon, where I discovered my passion for research. Later on, I decided to learn more about life sciences, which is why I obtained 2 master's degrees, one from the University of Guelph and the other from the University of Ottawa. I am interested in translational bioinformatics, and I have a genuine interest in cancer therapeutics. I worked at a hospital for 2 years during my first master's in cancer research, where I learned several basic research techniques and the introductory material and advanced material related to cancer biology. Cancer is an extremely heterogeneous disease. It's so vast that the research is still ongoing to solve this problem. We're learning more and more about how complex it is with greater molecular diagnostic techniques. To show you how complex cancer is, let me give you an example of my master's project. So there are several proteins that play different roles depending on its context. And the protein that I studied is called ST20-like kinase, or SLK. It plays an important role in processes such as proliferation, migration, and apoptosis. It affects embryo development and is shown to play a role in tumor genesis. This was studied in a new induced mammary tumor model within mice, and SLK knockout was shown to develop tumors faster and showed an increased expression of SOX10, an important biomarker in cancer. However, in my own research, which is an extension of the previous research, we studied SLK in mouse embryonic fibroblasts, where SLK knockout was shown to reduce focal adhesion turnover. It turns out that SLK plays different roles depending on its context. Even though this shows a different phenotype because of reduced migration versus increased tumor genesis in the previous study, this contrast shows how complex cancer can be depending on its context, and how we have to be very careful with the experimental design and data analysis to truly analyze its cancer's pathogenesis. Research on SLK is ongoing, which is similar to cancer. Every cancer is different, and it's a challenge to determine where those differences exist. We need more advanced methods to determine why a knockout reduces a process in one context but increases it in another. This master's thesis gave me more insight on what needs to be solved, and therefore I decided to do another master's in bioinformatics to learn complex analytical skills to solve these basic research problems. So therefore I transitioned from basic research to bioinformatics. Pure biology is good, but those data analytical skills can help us derive meaningful relationships to solve major problems. Bioinformatics has exposed me to a variety of other fields because of its interdisciplinary nature, such as statistics, math, and engineering. So my passion for biology and programming and teaching, because I used to volunteer and teach ESL to people in my community, and that slowly developed into a teaching career. Then I taught biology, SAT, and then programming, and it slowly developed. So my passion for biology, programming, and teaching eventually led me to Pine Biotech by meeting Ilya. So as an OmicsLogic's bioinformatics trainer for the precision oncology program, I enjoy going through these important pipelines, which can be relevant for my understanding as well as the learners. I personally believe that I've contributed to my learners' research when I answer their technical questions as a mentor. I enjoy working in the field of cancer therapeutics, and I'm grateful to share my passion for the field. Um, as a master's graduate in medicine and bioinformatics, I truly know the transition from basic research to bioinformatics. Therefore, I hope to make that transition as easy as possible for my learners. There are several courses on the OmicsLogic website that expands upon different topics within bioinformatics, and learning and teaching these courses helped me become a better bioinformatician due to the lesson preparation, which involves great understanding of the course material. Therefore, I look forward to teaching future OmicsLogic alumni. And I hope if you have any questions, please let me know.
So how could you recommend for somebody who's getting interested in oncology to get started with a research project?
So if I'd say you should do a needs assessment to see where your weaknesses lie. Usually if you're coming from a biology background, biology should be your strongest point. Therefore, I would maybe take some introductory to R courses or Python, just like there's some courses in the OmicsLogic website that pertain to the foundational technical knowledge to get into the field, as well as the introduction to bioinformatics course, which helps you gain those foundational skills to go deeper into bioinformatics when you have to deal with pipeline development and understanding why certain steps are important, like the heavy technical steps. I hope that answers your question.
Thank you, Clinton, for your wonderful presentation. We definitely have a lot more to learn from you. I would also like to thank all the presenters for today and the speakers as well for their valuable time and insightful information. I would like to pass it on to Beepsa ma'am for further insights into certain training programs. Beepsa ma'am, over to you. Thank you.
Thank you so much, Rutvi. And it has been wonderful to see all the presenters present their different research ideas and proposals. And on this note, as we have a lot of participants today who are interested specifically in research, I only want to take 5 more minutes of your time and continue with this presentation and also introduce everybody to some resources. So my name is Beepsha Biswas and I'm the OmicsLogic Community Manager. And I work with students, researchers, faculty from all different parts of biology, bioinformatics, biotechnology backgrounds. And what I have is an insight into how this entire process of research works. And I have been working with students like you, researchers like you for a while now. And what my main objective is to help anybody who comes into this community that we have, which is our bioinformatics community, achieve the personal goals that each person would have. So what I first wanted to talk a little bit about is the OmicsLogic programs. Now, what we saw presenters present today are some research projects. Now, as we understand that these research projects come only after you have gone through a certain degree of training. It could be any kind of degree training that you're taking at your university, college, or your school level, or it could also be some of the additional training programs. And what we do at Pine Biotech is offer training in multi-omics analysis. So what we try to teach all of our participants is, how you can work with different kinds of omics data. And right now we are working with thousands of users around the world. We actually have more than tens and thousands of users who are participating in programs like this. And we work locally with users in US, Africa, and India, where we are involved with universities, involved with local training institutions, and also with students who are joining in. This does not mean that other students are not joining in. We are also actively working with other students who join in on their own from different parts of the world. So these are some of our local chapters, as you can see here. And the next part is our educational program. So what we have is called OmicsLogic. So you can understand by the name is that these programs teach you about the logic of different kinds of omics data types. Now, I know that most of you are familiar with one or the other type of omics data, right? So maybe you can put down in the chat box, what are the different omics data types that everybody here knows? So please put those omics data types in the chat box and I can take it from there. So maybe Rutvi, you can put in one name to start with and also encourage everybody else to put in their comments. And what we actually hope for is that, as part of all of these discussions that we have every week in the club, all of the participants are able to participate in these discussions. So I hope, am I audible? Okay, Rutvi, okay, Pooja is writing epigenomics, great. Rutvi, genomics. Okay, Maham writes proteomics, wonderful. So you see, there are so many types of omics data that you all are familiar with. Damilola writes metagenomics, Sreyasee writes transcriptomics, perfect and wonderful. So we all are familiar with these different types of omics data, and that is exactly what we do with our programs. So we try to teach you the logic. So I will introduce you to some of these programs. For example, what Clinton showed you today, that he is training students who are in the precision oncology program, where we are talking about precision medicine for cancer research. Even today's topic was on cancer biology, right? So there we are trying to teach our students who are coming in and joining the program how they can utilize this particular kind of omics data to answer some of the questions that they have. And how do we do this? So you would see here highlighted in black that we have some interactive online tools. And I will also, again, introduce you to those tools in a moment. Now, what we do is that we first start by introducing you to these courses online. So every student who comes in for the first time, they are introduced to the courses. They start learning from the courses as well as online sessions that we have. And then we also introduce you to our research grade analysis platform. So this is a cloud-based platform called the Server BioInfo. And what we do as part of that platform is give you access. So each student has research access to that platform, where they are able to take data from publicly available data sets. For example, you saw that Rutvi has a data in mind. Now she will have to understand how to work with that data. So she also has gone through some of these courses, as is Sreyasee and Sraosee, who are also going through some of these courses right now. So they will be learning and then understanding whether on that data that they're looking for or the kind of analysis that they have to do. So this is exactly what we hope that we will help you learn, so that in the future, any type you are, say you have any kind of problem you want to solve using, say, RNA-seq data, or it could be a metagenomic data, you're able to do that. So we will be teaching you the logic. We will be working with you. You will have mentors here. We have Dr. Harpreet here, Ilya, Dr. Mohit Mazumdar, Clinton. And also there are other several mentors who are ready to walk you through these different steps on how to achieve that goal of where you also have a research project, and not only research project. So the next step is, once you complete these research projects, is like, presenting them. So here are just some of these examples of these research projects. And we take it to the next level where you can also complete the project and present them. So one of the things that we do as part of this club is, present a lot. We ask everybody to make sure that they have a presentation ready. They can speak clearly. Because when you have a research idea and you work on something, it's not enough if you just have that idea or you have worked on your project. You need to tell it to the whole world. So we go through with our research fellows, with our participants, so that they can present, they can prepare presentations, they can do video recordings of those presentations. So we walk through them, we correct anybody, whoever has, and we identify those gaps so that you become a better speaker, you can present them, and you can follow through a step-by-step process, which also I will be showing you very shortly. So these were just some poster presentations that we have had in the past. And we do hope that anybody else who is interested would be able to go forward and work through. Now, let me introduce you to the platform as well as some of the resources. So we have this platform called Learn.OmicsLogic. It is our educational platform. And what we have in this platform are a number of different courses. And the best part is that this platform is free. So you can log into this platform, and once you create an account, you will always have access. So you do not lose access to the platform. And if I'm to take a look at one of these courses, so say for example, genomics, you would find that there are several lessons. So many of these lessons are free, while some of these lessons are paid. So I would encourage anybody who is interested in learning more about any of these topics, be it genomics, be it metagenomics, or even transcriptomics, please go through. And there are several other lessons also here. So please feel free to look at those lessons as well and ask us any questions. You can put those questions down in the WhatsApp group that we have for SCI-OMICS, so that we are able to get back on the lessons with you. If you're already a part of our program, you know that we have Slack channels, you are there with us on Skype, as well as you have our email IDs. So please feel free to utilize those and ask as many questions as you can. So one of the things that, as I mentioned, that I've been working for a while with so many students, researchers, and everybody else. And one of the things that I feel is missing is that most of the time, students don't ask us questions, and then they wait till the end, and maybe then we say that, okay, you have to change this, and they're like, I have been thinking for a while. So what I encourage is that whenever you have a question, please ask us that question, so that maybe at that moment in time we are not able to help you, but we'll also go study a little more, learn from your question, and give you the right answer that you can then use to learn from, to maybe correct your project or any other thing. So it is a learning experience for everybody involved in the process. And I hope that this would give you an idea of how things are working. So definitely these courses are available for everyone to take freely. So please sign up to the platform. It's a very easy way to sign up. I will show you out. So let me log out here quickly and show you how to log in. So once you go log in here to the platform, you would come to this page. So you can either create an account, so mine is already saved here, or you can also utilize any of your social media logins to log in. So I already have my email ID saved up. So once I sign in, I'm directly brought to the course page. So you can continue to do this, and also you can track your own progress. So if I click on my email ID, I'm able to see what I have done so far. Right? So I think it's a very interactive way for you to learn. I also encourage you to go ahead and update your profiles here. You can see your activity on different dates. So please feel free to test yourself as well while you are going through that process. And the next thing that I wanted to just introduce everybody to are the projects. So we heard today about some project proposals as well as project ideas that our research fellows, campus ambassadors have here for what they want to do in terms of cancer biology or cancer research. Here are several different projects that have been done and completed by students who come from high school, like Amelia. And I think today we have both Dr. Gansha and also Dr. Chupuka in the chat box. So hello, Dr. Chupuka. So Dr. Gansha and Chupuka had collaborated on this project on SARS-CoV-2, and you can also watch this project on YouTube. So all of these our YouTube links. You can directly watch them. And here we have another project on triple negative breast cancer. So there are different projects, not only related to cancer, but on, for example, on neurodegenerative diseases, on metagenomics, etc. So we'll be continuing to update this page with any other projects that our research community completes and puts forward. And I do encourage that everybody who is present here today is also going to go ahead and propose their own project, work on their own project. And anytime you would need any kind of assistance, please feel free to reach out to us. And we can be reached at marketing@pine.bio for any kind of questions regarding how to approach a project, or if you have any specific concerns related to the courses. And the next part that I would want to introduce you to is the OmicsLogic program. So as I mentioned, that we have OmicsLogic programs on different topics that go on throughout the year. So at different points of the year, different months, we take up a lot of these programs. And now that we are at the end of an academic year and summer is starting, I believe that many of the students would be finishing their exams in their colleges, universities. So it's a good time for you to focus on something that would help you to learn more or advance more and learn more about bioinformatics, if that is an area that you're specifically interested in. So this is the link where we keep on updating about any of the programs that we have. And please feel free to ask me any questions that you may have. Of course, the first one is the research fellowship. This is a program that we offer all around the year for any student who is interested in working on their own novel research project. So again, to note, I use the word novel because the research project idea is actually proposed by the student. So we do not propose that idea. We do not make you work on our project. You propose that idea and we work to train you first. And the second step is asking you to present your own project. Like you see that Rutvi has done today, she's presenting a project idea. Then we work with you to finish that idea. Like it could be that you are answering a very broad question. So we would try to do something very simple so that you can start by looking at a problem, maybe that, a research paper that you have read must have missed out on, or there is a gap in that research. So we will take up those points and work with you to answer those questions. So this is like for 3 months or 6 months, you can apply for a research fellowship. Then we have a workshop in collaboration with the All India Institute of Medical Sciences, New Delhi. And this would be a 2-day workshop starting on April 23rd and April 24th. So this is also a workshop that you can sign up for if you're interested in learning more about lymphoma. So lymphoma is the topic that we are going to cover in this workshop. And third is this program that we are running in collaboration with the Noguchi Memorial Institute of Medical Sciences in Ghana. And the focus of this is on transcriptomics or RNA-seq. So this program specifically starts on the 16th of April. Yesterday, we had a free webinar, which is now available for everyone to watch on our YouTube channel. So please feel free to watch. And if you want to request a video, you cannot find it, you can again reach out to me at marketing@pine.bio, and I will be able to share with you the video of this session. So we are going to be starting this 1-month training program on April 16th and really work on focusing about infectious diseases. So talking about malaria, COVID-19 mainly as part of this program and other infectious diseases. We also have other RNA-seq training programs which talk about more about cancer biology or any other applications of RNA-seq to different kinds of biological problems. And the next program that we have upcoming is on biomedical drug discovery. So this is a joint collaborative program with Pine Biotech, Pharma Toppers, and a few other collaborators. And this will be a 4-month program. And what we are doing here is we are going to be taking again intensive, small programs that we are going to do, starting with genomics, then transcriptomics, then R and Python and structural biology. So if you're interested in either of these programs, please feel free to fill out the form. And at the end, you can choose which program you're interested in and then click on submit. So then you would receive the details regarding this program. So please feel free to ask me any question that you have about any of the topics that I spoke about today. And what I wanted to show you is an interesting thing of how we plan our research programs. So as you saw that projects, and as I've been speaking about the projects which are completed. So what we do is we follow this research process, right? Where we start first by the training that we are covering in the programs and the courses. The next step that a participant has to do is identify a topic by conducting literature review. Then identify a data set. For example, today, Clinton asked Rutvi, why did she choose the data set that she has chosen, right? So you would need to understand why you have to choose a certain data set. Why does it align to your project idea? So that is quite an intensive process. And therefore, we are here to be your mentor and guide you with that process in case you are not able to identify that. Then the next step would be, once you have identified the data set and everything, to prepare a table of metadata that would have the names of the samples, group names, etc. Then the next step is to present 3 to 5 slides of the idea that you have. Like we saw Rutvi present. She has an idea right now on how to work on medulloblastoma, but she's not very well defined. The idea is not yet defined. The idea is there. She has a certain interest in that topic. She has found some data sets, but she still needs to identify what is the research topic that she has, right? So these are how we move through. So there are these 20 steps that we are following through, and it's a very step-by-step and defined process. And as you see, most of the participants who have followed through these steps have had this outcome where they are able to present and then translate it. Some of our participants that we will be updating here are also going to have poster presentations. They'll be speaking at conferences. So you can imagine that once you have a project done, you can take it from to anywhere. So it's up to you how much research you want to do. Some of the times our participants are very happy completing a project and then applying or adding it to their CVs. And it's of course very helpful for career growth. The other times they're also interested in pursuing further research in the topic and identifying any other gaps that they may have in the answer, in answering the question that they were trying to answer, and go and research for it for years as well. And then you can present it, you can attend some seminars where you can present it to a larger community and not just a very localized community. And what we will try to do is help you present it in our meetings. So we continue to have these webinars, we continue to have these meetings that we have as part of SCI-OMICS Club, and we welcome you all to join this from time to time. And now again, if you have any questions, please feel free to ask those questions to me. And if you do not have any questions, then thank you all again for joining today. It was wonderful to listen to all the presenters, and I do hope to see more participation from everybody in this club. And again, please feel free to reach out to us anytime. Thank you so much. Thank you, Rutvi. And thank you everybody else for participating in today's session.